Hello, in this video we will understand about the enteroendocrine cells and gut hormones in metabolism during the fat state and fasted state. Remember, the enteroendocrine cells are present in the stomach and duodenum, is responsible for the secretion of the hormone endocrine hormone for the regulation of appetite and satiety or hunger and the feel satiety. So the key role in the control of metabolism we will understand the intestinal function due to the enteroendocrine cells, insulin secretion due to the enteroendocrine cells, nutrient assimilation into the cell, into the tissue, and food intake. Regulation. Gut hormone generates signals related to the nutrient absorption. So the absorption of the nutrients is due to the and feel the relaxation, increase the parasympathetic nervous system while inhibit the sympathetic nervous system. Sympathetic nervous system activate during the fasted state, the early fasted state. Here is when food will be intake. Go into the esophagus, stomach to the duodenum. This is the wrong. It is my mistake. The duodenum should be stomach. Why? This is stomach. Let's begin to understand the wall of the stomach. Contain a cell. The cell is known as the epithelial cell, but we will understand collectively the duodenum and stomach. So it's contain enterocytes. The brush border cells contain brush border enzyme. Enteroendocrine cells are also present. A lot of cells, the delta cell, the alpha cell, a lot of cells are present, but we will understand about the general quick overview about the fat and fasted state and how the regulation of the digestion. So the glucose, lipids, and amino acid when increased into the lumen after the digestion will should be absorption, but the absorption by a different mechanism. The C8 containing fatty acid when increased into the lumen and amino acid as well as glucose will enter into the L cell. This is the L cell and it is the enteroendocrine cell. When the free fatty acid and glucose will be enter, it will lead to glucagon-like peptide 1 L cell, which that will lead to exocytosis of the GLP-1. The GLP-1 will be exocytosis go into the blood circulatory system. It is known as the glucagon-like peptide 1. This is the peptide hormone. Whenever it will be going to the blood circulatory system, the K cell also work. The K cell contain a GLUT1 transporter channel, which that will lead to glucose influx. After the influx of the glucose, the GIP vesicle will be exocytosis. GIP means the glucagon-like insulinotropic hormone. While the enteroendocrine cell is here X and A cell, we will discuss later in this video, but first of all we will understand about the L cell and K cell. The L cell release the GLP-1 while the K cell will release through an influx of the glucose into the K cell will lead to GIP vesicle exocytosis into the blood circulatory system. Whenever the exocytosis of the GLP-1 and GIP will be change the game in the blood circulatory system, and in this way it is the identifier for the relaxation to increase the parasympathetic nervous system rest and digest, while the sympathetic nervous system increase due to the fasted state, early fasted state. The GLP-1 and GIP increase will lead to target to the pancreas. Pancreas contain beta cell, alpha cell, and delta cell. But we will understand about the beta cell because it is the target beta cell. The beta cell contain insulin containing vesicle, while here is the GLP-1 receptor and GIP receptor. Whenever it will be bind the GIP-1 and GLP-1 and GIP with a different receptor will lead to cascade of phosphorylation to enhance the cyclic AMP, as well as the glucose should be increased into the cell intracellular will lead to increase the ATP due to the glycolysis, Krebs cycle, electron transport chain in the mitochondria. Cyclic AMP increase, also the glucose due to the increase will lead to also glucose increase will lead to increase the calcium outflux from the endoplasmic reticulum. So here is the vesicle. Vesicle also cyclic AMP will trigger the DNA to more synthesis of the insulin vesicle for exocytosis further. And in this way, the increase the vesicle expression will lead to appetite again and again, will lead to cause the type 2 diabetes mellitus. And in this way, the damage of the beta cell also can be occur. Inflammation can occur. So we should prevent the snacking. Here is the increase of the insulin due to the exocytosis by the mechanism for the GLP-1 and GIP triggering mechanism to increase insulin. Let's begin to understand first of all the enteroendocrine cell X and A cell. Whenever the glucose will be bound with the G proteins, G coupled protein receptor, the ghrelin here is a fat state will lead to ghrelin inhibition through acylation of the ghrelin. After all, the beta oxidation will occur during the fasted state. This is the fat state to increase cyclic AMP also will inhibit the ghrelin through acylation of the ghrelin, and in this the hungry will be decreased, the hunger will be decreased, appetite will be suppressed, while the fasted state later on will lead to increase the long chain fatty acid, and the long chain fatty acid and short chain fatty acid will lead to fasting to produce beta oxidation in the mitochondria to produce inhibit the ghrelin to decrease the appetite later on. And sympathetic nervous system activate the early phase of the fasted to produce norepinephrine will lead to inhibit the ghrelin later on, not earlier. And the ADRB1 should be trigger, and the CASR will also inhibit, as well as the free fatty acid 4 domain will also inhibit. Due to the long chain fatty acid increase due to the lipolysis and the beta oxidation, and amino acid increase due to the proteolysis, not proteogenesis, during the fasted state. Let's begin to understand the ghrelin and leptin. The ghrelin is the far side note, the stimulatory effect of adipogenesis, ghrelin release, and hunger hormone it is, while the leptin is against the ghrelin and satiety can create. So the leptin will release due to the lipolysis to inhibit the hunger, while the hunger can stimulate through ghrelin, so the ghrelin should be inhibit. So in this way, here is the tissue which that's contain insulin receptor, tyrosine receptor kinase. In this way, when bind the insulin, when increase the insulin is known as the hyperinsulinemia due to the hyperglycemia, when the absorption of the glucose should be assimilate into the cell, and this is the DNA. Also, the insulin receptor with cascade of phosphorylation lead to expression of the gene for increase the GLUT4 vesicle, will transport into the membrane, will increase the poration to allow the glucose influx into the tissue, into the cell. Let's begin to understand the summarization. After the assimilation, the complete will be enteroendocrine cell mission will complete. The summarization is that the fat state and fasted state. The fat state, the glucose, amino acid, and fatty acid will be increased into the lumen, and then after absorption into the blood circulatory system should be assimilate due to the GLP-1 and GIP-1 GIP increase. While the fasted state will lead to increase the beta oxidation of free fatty acid, ketogenesis to produce ketone, and gluconeogenesis. After all, the ghrelin, the hunger, will lead to inhibit the ghrelin, and in this way the beta oxidation of fatty acid and ketogenesis will also inhibit the ghrelin, while the early fasted state will trigger. This will lead to GLP-1 and GIP will decrease the downregulation of the beta oxidation of free fatty acid and other due to the fasted state, while increased during the fat state. And this basically lead to assimilation. The GLP-1 and GIP is responsible for the assimilation due to the production of insulin. After this assimilation, the ghrelin will be triggered after the complete assimilation lead to again appetite. While the GLP-1 and GIP also trigger the pancreas will decrease due to the fasted state, so the ghrelin inhibition will occur. And in this way, the parasympathetic nervous system increase. Thanks for watching. If you like my video, please make sure to subscribe, like and share, and give the comment below for understanding better if you have any doubt. This video was about the enteroendocrine cells and gut hormone in metabolism during the fat state and fasted state for the regulation of the digestive system, regulation of the digestion. So next video we will understand about the further exploration about the working of the insulin and how glycogenesis will occur, lipogenesis will occur, and how the leptin and ghrelin can interact to each other. So we will also learn about the working of the leptin and how it's released